For the introduction of high-performance lipid chromatography, or known as HPLC, it is a laboratory technique that allows a very efficient separation of small amount of components of a mixture. It utilizes the liquid mobile phase to separate components of a mixture. The analytes are first dissolved in a solvent and then forced through the columns. Columns in HPLC contain tiny particles about 5 micrometers, and the solvent that were forced through the column are under very high pressure. After it travels through the column, the detector and the computer that are connected to the HPLC instrument record the data and sense the signal when the eluents are coming off the columns, and the fraction should be collected. The technique is actually very similar to an operational basis of lipid chromatography, but HPLC allows a far better separation of components of a mixture. And now, for the key principle in HPLC, first it begins by the injection of solid into the column, and then the separation occurs as the analytes and mobile phase are pumped into the column. Each of the components elutes from the column are recorded as peaks on the computer. And lastly, the response of the detector to each component is displayed and known as the chromatogram. There are three objectives for the experiment. The first objective is to determine the retention time of a standard solution, which is caffeine. For the second objective is to identify the caffeine peak in a soft drink sample. And the last objective of this experiment is to determine the amount of caffeine in a soft drink sample using the response factor method.
For the methodology, this is the fully labeled block diagram for HPLC. The flow in order starts from solver reservoir to degasser, pump, injector, guard column, analytical column, detector and waste collector and data acquisition system. There are several components in HPLC and each of them have their own functions. The solvent reservoir is a container that holds the mobile phase before it is pumped into the system. The degasser removes any dissolved gases and dust from the mobile phase. The mobile phase are then forced through the system at high pressure and constant flow rate by the pump. After that, the re-injector will introduce the sample into the mobile phase. And the guard column protects the main column by trapping any contaminants and particulate matter. Next, the analytical column interacts with the mobile phase to separate the components in the sample. The detector then detects the separated components as they exit the column. After that, the mobile phase and eluted compounds are collected in the waste collector after they exit the detector, and the data from the detector are collected by the data acquisition system to be processed and displayed in the form of a chromatogram.
Now, we will examine the results of a caffeine content analysis using HPLC. We will compare the caffeine levels in a standard caffeine sample, a sample of Coke, and a sample of Levita. First, let's look at the first result, the standard caffeine sample. The chromatogram clearly shows a single, well-defined peak corresponding to caffeine. The area under this peak is directly proportional to the amount of caffeine present. The next result, we have the Coke sample. Again, we see a prominent caffeine peak, which is the fourth peak that I have circled. However, there are also other peaks present, indicating the presence of other compounds in the Coke. This is expected as Coke contains more than just caffeine. Finally, let's look at the Levita sample. Similar to the Coke sample, we observe a main caffeine peak, which is the fourth peak, along with other peaks representing other components. Table summarizes the HPLC results. The compound column lists the sample. Retention time indicates how long it took for caffeine to pass through the HPLC column. The peak area is directly proportional to the amount of caffeine present. Let's look at the result. The standard caffeine sample had a retention time of 2.482 minutes with a peak area of 6,249.49. The Coke showed a retention time of 2.481 minutes with a peak area of 7,001.12, and Levita showed a retention time of 2.479 minutes with the highest peak area at 11,151. And note that the retention times are very similar across all samples, confirming that we are indeed measuring caffeine. In HPLC, we often use a response factor to relate the peak area of an analyte to its concentration. Now, we will walk through the calculation of the response factor for caffeine. Let's apply this formula to our caffeine standard data. we have a peak area of 6,249.48 and a concentration of caffeine standard 0.1 mgml. Substitute this value into the formula, we get the answer of 62,494.87 mgml. This is our response factor for caffeine under this specific HPLC condition. Therefore, the response factor for caffeine in this analysis is 62,494.87 mgml. This value is specific to the HPLC method used, and the detectors respond to caffeine. This RF is then used to calculate the concentration of caffeine in unknown samples based on their peak areas. To calculate the caffeine concentration, we will use this formula. Concentration in parts per million ppm equals the peak area of caffeine in the sample divided by the response factor. Let's start with Coke. We have a peak area of 7001.12. Substituting this value and the response factor into the formula, we get the answer of 0.1119 mgml. To express this in ppm, we multiply by 1000 ppm over mgml, resulting in a caffeine concentration of approximately 112.027 ppm in Coke. Now, let's do the same for Levita. Its peak area is 11,151.1. Following the same formula, we get the answer of 0.17843 mgml. Multiplying by 1000 ppm over mgml, we find a caffeine concentration of approximately 178.432 ppm in Levita.
High-performance liquid chromatography (HPLC) is an analytical technique where it is used to separate, identify, and quantify the elements of mixture. This analytical technique separates the molecules of the mixture based on how the molecules interact in stationary phase, where the solid materials are packed inside a column, and a mobile phase where a liquid solvent that flows through the column. The result or identification of this analytical method is by carrying out the retention time of the samples. In this experiment, 3 samples were run, which is standard caffeine, Coke, and Levita. For the first sample, standard caffeine shows retention time is 2.482 minutes and the percent area is 100%. The second sample that will run was Coke sample. The retention time obtained was 2.481 minutes and the percent area is 57.36%. Last sample that was tested is Levita sample. The retention time obtained was 2.479 minutes and the percent area is 17.70%. The peaks that were chosen to be the results of the sample is the 4th peaks because the retention time obtained is the closest to the standard solution reading, and this shows the accuracy of the results. The response factor of the caffeine solution was calculated by dividing peaks area of standard caffeine to concentration of standard caffeine. The response factor is 62494.8779 mg per ml. The concentration of caffeine in the unknown sample were also calculated. The concentration of caffeine in Coke is 112.027 ppm, while concentration of caffeine in Levita is 173.432 ppm. This shows that concentration of caffeine in Levita is higher than concentration of caffeine in Coke.
In conclusion, we were able to determine the retention time of a standard solution caffeine, which is 2.482 minutes. Besides that, we were successfully identified the caffeine peak in a soft drink sample. The peak area of caffeine for Coke is 7001.12305, while the peak area of caffeine for Levita is 11151.10000. Lastly, we were able to determine the amount of caffeine in a soft drink sample using response factor method, which are 112.027 ppm for Coke and 178.432 ppm for Levita. Now, I will explain about other applications of high-performance liquid chromatography (HPLC). The first one is for food safety. For example, HPLC is widely used to detect and quantify pesticide residues in food products. It can identify trace amounts of pesticides that might be present due to agricultural practices, ensuring that food products are within safety limits set by regulatory bodies. Next, HPLC is also important in water purification. It is highly effective in detecting organic contaminants in water, including pesticides, herbicides, industrial chemicals, and pharmaceuticals. These substances may be introduced into water through agricultural runoff, industrial discharge, or improper disposal of chemicals. Not only that, HPLC is also commonly used in detection of drug traces in urine. When drug traces are detected, the HPLC findings can be used as evidence in police charge as well as sport disqualifications, since it can be used to check for metabolites produced by a reaction to the substance. It is useful for modern drugs, which are designed specifically to disappear within the body. Last but not least, HPLC is also widely used in manufacturing process of pharmaceutical products. HPLC is a reliable and precise way to check product purity and helps measure the exact concentration of the active ingredient in pharmaceutical formulations such as tablets, injections, and creams. As a result, it can help pharmaceutical manufacturers develop the purest products and ensures that each dosage contains the correct amount of the ingredients.